Here we go. All
That's
right.
my internet fell out. That's my fault. I'm
Oh,
glad the room didn't close.
no worries, no worries. All right. First off, I guess why do we study paleontology? So bats are incredibly abundant among living mammals, right? They're the second most abundant group. There's something like just shy of 1,500 living species of bats, and we can do things like look at the similarities in their DNA to understand how they're related. We can look at their development. We can look at all these things um, among extant bats, and the fossil bats uh, oftentimes look very similar to extant bats. So why do we study paleontology? Well, if we're not looking at the fossil record, we're only looking at a part, in fact, a small part of bat evolution. So on the right side, you'll see a general generalized tree of relationships of organisms, right? And um, for those that aren't maybe familiar with reading these, um, species A and species B are most closely related to each other because they share a point most recently in time that they were on the same line, right? So if you trace the lines back, they join up at that node. Same thing for D and E, and then F is most distantly related to everybody. So this is what we can see if we're looking at the extant, the living organisms, but there's a whole diversity of fossil organisms that fall into um, these relationships uh, in ways that we wouldn't know otherwise, right? So we have an entire group down at the bottom, those two red X's that we wouldn't know without the fossil record. We also have sort of um, species that are sister species. They're closely related to other members of this tree. F and C have closely related species that are extinct, and then we have sort of uh, uh, ancestors in the fossil record of E and this group A, B, and C. So this is broadly why we study paleontology. Um, and I guess for a little bit of context, what is paleontology? It's the study of fossil or extinct life, right? Um, so it's essentially the crossroads of biology, the study of life, and geology, the study of Earth and the study of time, deep time. So there are a number of different subsets of paleontology, people who specialize in studying fossil plants or invertebrates like crustaceans or insects. Um, People who study trace fossils, trackways, burrows. And then for this talk, we're focusing on a vertebrate group, a mammal group, bats. So this would fall under the umbrella of vertebrate paleontology. And there are many other subsets of paleontology. And then there are sort of more synthetic fields of paleontology that incorporate evidence from a lot of different avenues, things like paleoecology, that then can feed back into our understanding of vertebrate paleontology or paleobotany. So that's what paleontology is. Um, well, all right, one more slide of background, I guess. For those not familiar with the geologic timescale, um, what we're focusing on is the Cenozoic era. That's the modern era that we currently live in. It began 66 million years ago with the extinction of the dinosaurs and continues until today. And so when you read a geologic timescale like this, you'll read from bottom to top. So it goes oldest at the bottom and youngest at the top. And for instance, bats show up at the beginning of the Eocene about 56 million years ago and continue all the way until the modern. Uh, Sometimes you'll see time laid out on its side. And if you're unfamiliar with that, you'll typically read left to right. So again, bats show up about 56 million years ago and continue all the way until the modern day. So how do we recover fossils like this? So one of the sort of stereotypical ways of excavating fossils in the field is quarrying, right? This is what people think of when they think of going out and digging for dinosaurs. Often it's a little bit of bone that's exposed, and you sort of excavate with things from maybe pocket knives, like in the picture, or jackhammers, right? There's a whole huge range to remove this rock, expose these fossils, sort of prepare them and excavate them from the ground and bring them back into the lab. So this is really good for large fossils or locations where fossils are really, really abundant. Typically, this isn't how we recover fossil bats, but there are a few really unique localities that I'll talk about in a minute where quarrying does recover fossil bats. Surface prospecting is what I do most. So this recovers sort of fossils that are exposed on the surface, eroded out or almost eroded out of sort of soft sediment. You sort of walk around bent 90 degrees, looking at the ground until you see something, or you crawl around on your hands and knees. And this is really useful for identifying locations that you can quarry or that you can screen wash, which we'll talk about next. So it requires a little bit of developing a search image. On the top right, you can see this jaw fragment of an early mammal with two teeth at the very top of that bone there sticking out. But this is sort of the introduction to how we find sites to quarry or to screen wash. And then screen washing is the best way to recover bats because bats are tiny. So this is sort of like panning for gold. We go out, we find a place where we're pretty sure we can find fossils. We've identified it by surface prospecting, and then we collect a bunch of sediment and take it to a nearby river or lake and pour it through a sieve. And all of this silt, the really fine particles fall out, and we're just left with small grains and pebbles, uh, as well as fossils. And so we let that residue dry in the sun, collect it back up, take it to the lab, and then we sort through it under a microscope. And we can recover really tiny, millimeter-sized fossils through this process. So what do we have as far as the bat fossil record? I've talked a lot about the methods. What do we have? So when you think of fossil bats, a lot of people are probably going to think of these fossils. This is from the Green River Formation in Western North America about 52 million years ago. And these are beautiful, complete skeletons of three species. And this is one of those locations where you can quarry. In fact, there's a lot of people quarrying here for things like fish. They recover reptiles like crocodiles and turtles. But with enough persistence, you'll probably find some bats. Over a long enough time, quarrying enough, bats are at this locality. We know that. You may also be familiar with this other locality, the Messel Oil Shale in Germany, and that's about 48 million years old. And it's, again, another locality that preserves really nice, complete skeletons of bats, and in even greater numbers than the Green River Formation. So Green River Formation, there's a few dozen specimens of 3 species. Um, Messel oil shale, there are several hundred specimens of at least 7 different species. So this is sort of what people typically think of when they think of the bat fossil record. But these are not the only bats. In fact, this is not even the majority of the bat species that we have in the fossil record, because most bats look like this. Um, and this is sort of a trend in mammal paleontology. We look at teeth a lot. Mammal teeth are very specialized. And so we can look at these isolated teeth and say, these are bats. Um, and in fact, these are older bats than the bats at Messel and the bats at uh, Green River Formation. We have 54 million year old bats from India, 55 or 56 million year old bats from Portugal and China, and 54 and a half million year old bats from Australia. So this is a huge chunk of the bat fossil record that typically goes overlooked. And since I'm talking about teeth, I have to give you a little bit of background on mammal teeth, because that is a huge component of mammal paleontology. Placental mammals have very specialized teeth, especially molars. They're what we call tribosphenic, so they have 3 primary cusps on the upper molars. So those are highlighted in red. And those sort of are paired with lower molars that usually have 6 main cusps. And they fit together very precisely, precise dental occlusion, which means as mammals change, maybe they start to exploit a slightly different diet, or they're just speciating, they're just gradually diverging. Um, the teeth change, both the upper and lower teeth change, which means mammal teeth are very good for both understanding the ecology and understanding just the relationships of fossil animals. So we can learn a lot just by looking at isolated teeth or jaw fragments. They're also teeth are covered in enamel, which is the hardest substance in a vertebrate's body. So they're preserved very frequently. Um, so mammal teeth are very useful in mammalian paleontology, and bats themselves have a handful of sort of modifications to this general plan that allow us to recognize them in the fossil record. In the grand scheme of things, bat molars are still pretty generic for mammals, but they do have a few really diagnostic features. You'll see on the cheek side of the upper molar there, you have this W-shaped crest. That's very typical of especially fossil bats, but most living bats as well. And on the lower molars, you see also on the cheek side, you have this sort of shelf at the base of the tooth. And there are a few other features that we can look at and say, yeah, this is probably a bat because it has these traits. So we can use all of this evidence to understand the relationships of fossil bats to each other and broadly to living bats. So for the most part, our understanding of fossil bat relationships have relied on a handful of studies of these really well-preserved specimens. And so at the bottom, you sort of see this group of living bats, and all of these fossils that are known from either Messel or Green River sort of branch successively more distantly from the living group of bats. So in this case, Paleochiropteryx, that lowest image, is most closely related to all of the living bats, and then on and on with Onychonycterus, this bat from Green River, being most distantly related to living bats. However, more recently, we've started adding a lot of more fragmentary bats to these analyses, and we start to get a more complicated picture of how these fossil bats are related to living bats. So here's a really cool study that came out just last year. And you can see those same 5 bats highlighted on this image. Um, and again, you get the Onychonycterus, so the tree is sort of inverted here, but it, along with its close relatives, are branching very early, but then everybody else is sort of lumping together, most closely related to each other, rather than to the living group of bats. Those are the extant bats right about in the middle of the screen. So that's a really interesting situation that we wouldn't maybe necessarily have expected if we didn't include these more fragmentary bats that are represented by pretty much all of the species that aren't shown on the right. And then we performed another recent analysis that just came out a few months ago, that found similar results. So again, most of these, in fact, in this analysis, all of these fossil bats are coming out more closely related to each other than they are to the living bats, and that's that last very bottom branch that's going off to the right that says crowned bats. So um, the other thing that we recovered in this analysis is that many of these fragmentary bats are branching earlier than some of these Green River and Messel bats, which is also fairly interesting, and hints that these fragmentary fossil bats probably have a lot to say about early bat evolution. So that's what we can do with understanding how bats are related to each other, uh, with sort of these fragmentary fossils, especially teeth and jaw fragments. But we can also start to understand what these bats were doing. What were they eating? And so on the right, you see a bunch of drawings of living bat dentition, um, and divided up by bats that we know are primarily eating insects versus primarily eating vertebrates, things that eat both plant and animal matter, or frugivores or nectarivores. And you see changes in the shape and the size and the number of teeth. So by looking at living bats and understanding how their teeth shape changes, we can start to apply that to the fossil record. And for instance, on the left here, we have a number of fossil bats, a generalized, or a bat that we think is a generalized insectivorous bat, in A, and so you can compare those teeth with what you see in A on the right. A bat B that we think is maybe a carnivorous bat, a bat that's feeding primarily on vertebrates, and so you can compare B in each figure to each other, and then a bat that we think is probably a so-called dedicated omnivore, and so you can compare C to each other and start to see some of the similarities in how these fossil bats are changing the shape of their teeth, um, to fit their diet. But there are other analyses of other bats that sort of maybe result in more surprising results. So this is a really cool study from a couple of years ago about this bat, from about 13 million years ago in South America, called Notonycterus. And Notonycterus is thought to be particularly closely related to the spectral bat and the woolly false vampire bat. So these living bats that we know are big specialists on terrestrial vertebrates. So they feed primarily on terrestrial vertebrates. Um, and this study looked at the shape, in this case, of the lower molars. They approached it in several different ways, but they looked at how these lower molars compared in shape to living bats that we know feed on vertebrates or fruit or insects or are omnivores, um, to see where what this bat was most similar to in terms of tooth shape. And so in the center of, for instance, A, you can see that gold diamond, and that's Notonycterus. And it's surrounded by that green box, which represents the space occupied by all of the insectivores. So it's very similar in shape to insectivore diet, um, or insectivore teeth. Uh, it's also just outside of the space occupied by the omnivores. Those are in the blue squares. It's relatively far from the carnivorous bats, things like the spectral bat or the woolly false vampire. So based on this study, we think maybe Notonycterus was more of an insectivore or an omnivore than a dedicated carnivore like its close living relatives. So this is the type of thing that we can start to understand with just with the dentition of bats. But we obviously don't just have a dentition. We often also have, or at least in some of these really well-preserved bats, we have postcranial bones. And we can start to understand how these bats might have flown and foraged using these postcranial bones, in this case, wing shape. So by comparing to living bats, we can tell that some bats have long, narrow wings. Those are what we call high aspect ratio wings. Some bats have shorter, broader wings, and those bats fly and forage in different ways. Long, narrow wings are good for flying very fast, but they're not particularly maneuverable, whereas short, broad wings are much more maneuverable, but they're not as good for fast flight out in the open. So on the right, you'll see a number of fossil bats that are known from complete skeletons and how their wing shape is reconstructed based on the dimensions of the bones. Um, and you'll start to see some of those same differences in wing shape. So for instance, Icaronicterus index right at the middle of the screen, you see has a sort of short and broad wing, much like image C of the living bats. Whereas something like Hesianicterus mesolensis has a longer, narrower wing. So we think these bats are probably foraging in different places within the forest, and exploiting different habitat niches. Um, so that's great, but like I mentioned, a lot of fossils aren't these really nice complete skeletons like this. Um, Isolated postcranial bones are or have been studied much less frequently. Um, on J, you can sort of see this or the schematic next to image J, this image of early or a recent early fossil bat that was described from a number of fragmentary postcranial bones. This one's nice because we can sort of use that and compare it to those fragmentary bones in other bats. So for instance, down at the bottom right, there's a distal humerus, which tends to be, so that's the upper arm bone, that tends to be the most or one of the most informative bones of the postcranial skeleton. Um, and in fact, a really cool study um, about a little more than a decade ago, um, looking at the distal humerus of this fossil bat from about 20 million years ago in Australia, called Icorops. And Icorops is thought to be closely related to the living New Zealand short-tailed bat. And New Zealand short-tailed bats are super cool because they're probably the most terrestrial of living bats. They spend a lot of time on the ground, sort of foraging for insects and feeding on pollen and nectar. Um, and they have morphological adaptations. They have special bones and things to allow them to forage in that way. They tend to have a really flexible elbow joint, for instance. So this fossil bat that you see on the upper right has similar adaptations on the distal humerus. So that's the part of the upper arm bone that contributes to the elbow as a living New Zealand short-tailed bat. These bats have these adaptations well before their lineage was isolated in New Zealand. These bats, Icorops, are from Australia. So this is some evidence that these bats were already capable of terrestrial locomotion before they were isolated in New Zealand, and that maybe that was just lucky that they could exploit that terrestrial mammal foraging niche when they were isolated in New Zealand, because they had those abilities for some reason when they were in Australia. So the other thing that makes bats, aside from flight, really, really bizarre among living mammals is the ability to echolocate. And we have this evidence in the fossil record as well, especially when we have skull bones in some degree of articulation. Um, the cochlea, which is sort of the last step before sound is transmitted to the brain, is very enlarged in echolocating bats compared to non-echolocating bats. So on the right, you see this really enlarged, spiral-shaped cochlea for an echolocating bat, and it's actually not even visible in the image for the non-echolocating bat. There are also, among living bats, differences in cochlea size and shape among echolocating bats. So there's a lot of potential for understanding the echolocation ability of living or of fossil bats by studying the size of the cochlea. So one interesting study that focused on this was on this bat that I've mentioned a couple of times already, Onychonycteris, which, based on the skeleton, is the most primitive looking bat that we have yet discovered. Um, and it also has, for its skull size, a fairly small cochlea. So in this chart, this graph here, you can see cochlea width. That's a proxy for the general size of this structure, to basicranial width, which is again a proxy for the size of the skull. And you'll see that non-echolocating living bats, those are the triangles, have a general smaller cochlea relative to the size of the skull than the echolocating bats in blue do. And Onychonycteris falls along that line of non-echolocating bats and outside of the region of most echolocating bats. So this is evidence that Onychonycteris may not have been able to echolocate, at least not nearly to the degree that most extant bats are able to. So whereas most of the other fossil bats indicated by the dots, we're pretty certain we're capable echolocators. So we're able to understand the ability of things, bats to do things like echolocate based on structures that can be preserved in the fossil record. And we can sort of synthesize all this information to understand um, early bat faunas. Just like today, we frequently don't find um, just one isolated bat in a locality, right? Today, we don't, other than very cold or remote places, usually there are several bats or dozens of species of bats occupying different roles in the environment, exploiting different parts of their habitats. And we can observe the same thing in the fossil record. In this case, you see two of the really great bat faunas, the faunas that we know complete skeletons from, and we can sort of understand how bats are partitioning out those environments by things like changes in the shape of their teeth, wing morphology, gut contents, which I didn't spend a lot of time talking about or spend any time talking about really. But we do have from some of these really well-preserved localities, and then by understanding how they're able to echolocate. So we can look at something like Messel and look at Tachypteron and say that bat was probably flying very fast. It was flying above, foraging above the canopy, versus something like Paleochiropteryx, which is not a fast flyer, but very maneuverable and was probably foraging within the understory around all of this vegetation. Um, so this is sort of how we study what bats are doing in the past in a paleontological context. Um, but with bats, there's this big question of where did bats come from? And it's one of the main questions in mammal paleontology and mammal evolution because bats are so specialized, they're so bizarre, and we don't have a good handle on where they came from. So I'll just talk briefly about what the fossil record can tell us about bat origins. So, unlike most other mammal groups, especially the sort of well-studied mammal groups, bats don't have obvious transitional forms in the fossil record. So we don't have anything that we can say, all right, this is half bat. We also don't really know what might be their close relatives in the fossil record. Unlike these other groups, so carnivorans or ungulates or primates or rodents, we either have very early members of those groups relatively soon after the end Cretaceous extinction, or we have things that look like they're probably closely related in some way. With bats, we don't have that. And when they show up in the fossil record, they look pretty much like bats. So we can start to parse this out by understanding what they're most closely related to among living organisms. So bats belong to this group called Laurasiatheria, which also includes the even-toed ungulates, so things like cows and deer and whales, the carnivorans, cats, dogs, bears, the group including shrews, moles, and hedgehogs, pangolins, and the odd-toed ungulates, so horses, rhinos, and tapirs. This doesn't necessarily help us in understanding the body plan of these early bats or what the earliest bat relatives would look like, because all of these groups are very diverse and distinct in morphology, and their earliest relatives also seem to be pretty diverse and distinct in morphology. So that doesn't necessarily help us in a paleontological context trying to understand where bats came from. But um, we are pretty certain that this group traces its origins to the northern continents. Those are Asia, Europe, and North America. And so um, this gives us a bit of a place to start if we're trying to understand what fossils or where to look for particular fossils that may be closely related to bats. So this is what the Earth looked like more or less when bats first show up in the fossil record. And it's not a great image because there would have been a connection between North America and Asia via the Beringia region, so very far northeastern Russia and Alaska, and also a connection between North America and Europe via Greenland. So these northern continents were all connected, and mammals and animals, plants were able to travel between these continents, especially in particularly warm periods. Whereas the southern continents, South America, Africa, India to some degree, Australia, Madagascar, and Antarctica, were all separate. So the fact that all of these other groups, the ungulates, the carnivorans, were only found in the northern continents in the fossil record suggests that since bats are very closely related to them, that's where we can look for bat relatives. And that's really nice because really quickly after they evolve or after we first see them in the fossil record, bats get everywhere. So the fact that their origins can be traced to the northern continents gives us a little, narrows down that area where we should be looking a little bit more. So what do we think might be related to bats in the fossil record? There's no real good answer. This is a group that I've been interested in recently that have frequently been confused for bats. They're only known from teeth and jaw fragments, like many early mammals. They're called nyctotheres, and that name means bat beast. Often, members of this group have been thought to be bats. So on the right, you can see Wyonycterus, which was originally described as a bat, and now we think is probably a nyctothere. But Cambea on the left is a bat and was originally described as a nyctothere. So there are some similarities in the dentition, in the teeth shape of these animals that suggest maybe they're related to bats. We do, based on recent analyses, think they're probably Laurasiatheres. So another place to start, we do only find them on the northern continents, and there's one species that we know from India. So maybe this is a start to understanding where bats came from, but that work is still ongoing, and we still don't have any great candidates. And what were these early bats potentially doing? So there has been a thought that the transition to powered flight may have passed through a gliding phase for these early bats. And this is a really cool study from a few years ago that looked at this most skeletally primitive bat, Onychonycterus, and removed the hand wing to sort of see where it fell with relation to living flying bats and living gliding mammals. And it actually is very similar. So AR represents aspect ratio, that's long and narrow wings versus short and wide wings, and WL is wing loading, which is a relationship between the size, the body mass of the bat or the animal, and the area of the wing. And without the hand wing, without the membrane between the digits, this this early bat looks very much like the sort of most specialized living gliders. Um, Whereas with the hand wing, it's very close to the space of living bats. So this might be the sort of key innovation that allowed bats to go from tree-dwelling gliding organisms to sort of flapping, powered flying bats. So um, this hand wing is a very rare thing among living gliders. In fact, there's only one living glider that exhibits this um, hand wing, and it also happens to be the most proficient living glider among mammals. Those are flying lemurs. They're not particularly closely related to bats, but perhaps a sort of comparison for what the flight membrane of these earliest bats might have looked like. So, why study bat paleontology? Hopefully, we've answered this question. Um, it provides us a more complete picture of bat evolution. So we know of entire groups that are now extinct that have something to say about how some of these traits developed. It helps us understand both the morphology, the shape of these early bats, but also the behavior, the ecology, the diet of early bats, how they were partitioning their habitat, how they were exploiting various resources in their environments. And it's vital to understanding bat origins because at some level, the transitional bat is not preserved. Like we don't have a living transitional bat. So if we hope to see the steps that bats took to become these very specialized flying, echolocating, um, diverse, successful organisms, we have to look in the fossil record because that's where we'll find that missing part of bat evolution. Um, so I think that's what I have. A lot of the studies that I mentioned in this talk, especially for the YouTube video, I'm flashing up on the screen here if you want to check them out. There's obviously many, many more. And with that, I appreciate your attention and thanks for having me.
Yeah, thank you so much, Matt. Do we have any questions from the gallery? Well, I have one.
Yeah, yeah, yeah.
So thinking about how it seems like a lot of the evidence is pointing towards the evolution of bats from the fossil record happening in northern continents.
Mhm.
And yet today, most of the biodiversity that we see does seem to be concentrated in the global south.
Mhm.
So what are what is your knowledge or what are your hypotheses as to like why we see this transition from the evolution occurring in these more northern latitudes and now seeing the biodiversity in further south?
Yeah, that's a really good question. And I think it goes to the fact that currently in our sort of world, climatically, we're in an ice house world. I know we're warming rapidly, but we there are polar ice caps that currently exist in both the far north and the far south, and very sort of seasonal environments in most of places like North America, Europe, Asia, um, and not so much in the South, really. So most of the tropics are concentrated in those regions today, which are places where you can have bats that live year round feeding on nectar and live year round feeding on fruit, and exploiting the huge diversity of, for instance, insects or other arthropods that also exploit those nectar and flowers and fruit, etc. And so in the early Cenozoic, in around the time that we see the first bats in the fossil record, we have an event called the Paleocene Eocene thermal maximum, and that's a huge spike in temperature. Actually, the whole early Cenozoic is a hothouse, so we don't have ice caps, and we have at points, including the Paleocene or Eocene thermal maximum and the early Eocene climatic optimum, which we do know we have bats definitely by that point. They show up in the fossil record right after the Paleocene Eocene thermal maximum, and they're definitely there by the early Eocene climatic optimum. We get things like primatomorphs and alligators in what's now the Arctic.
Hmm.
So places like Wyoming, where we have these early well-preserved bats, things like Onychonycteris and Icaronicteris, it was at the very least a sort of subtropical, uh, uh, very nice habitat, I guess, for these early bats, pretty much year round. So I think that's sort of why, like, they were able to evolve in the northern continents, and that is also just where their ancestors, the ancestral Laurasia-Therian was evolving in the northern continents because it wasn't a flyer and couldn't cross oceans to get to the southern continents. Um, but also the habitat was very suitable for them during the early Cenozoic. That said, we don't have a lot of fossils from this time in the global south, especially in existing tropics. They're not great for preserving fossils or for finding fossils, right? There's a lot of ground cover there. Um, and I would guess that bats probably radiated um, once they reached places like Central Africa or South America as well. They probably took off just like they do today. We just don't have a great record of it from the a fossil standpoint because it's hard to find those things.
Cool. All right. Do you have any more questions?
I have
Hey,
a question.
Oh.
No, Nancy, go ahead. Go ahead.
Okay, my question is, if what fossil gap or fossil place in space and time would you most like to see filled? Like, what would be the most exciting new fossil find you could imagine right now?
Oh, I really want a late Paleocene, so just before bats show up in the fossil record, um, a late Paleocene Lagerstätten, like a beautiful deposit, like things like the Green River Formation or Messel, um, on one of the northern continents. I'm really interested in Central Asia. I think Central Asia, a lot of cool things are probably happening there, but I'd take North America, Europe, Asia in terms of trying to understand where bats came from, because some of these little I focused on nyctotheres because that's what I've been focused on a lot recently. Um, but a lot of these insectivores from the early Cenozoic, we only know from teeth and jaw fragments, and it would be really interesting to see some of the rest of the skeleton of these things, um, the type of preservation that we could potentially get if we had a really nice deposit like Green River or Messel that was just a little bit older than when bats first show up in the fossil record. So that's what I really want to see. I don't know. What about you, Nancy? Oh, all right. Well, we'll let Heather. Oh, go ahead. Oh, whoever.
All right. Ready for me now?
Yeah, sure. I'm glad you
Right
showed
here.
up, Heather. Hey.
Also, I have one of your like original classmates with me here.
Oh, hey, Walter.
We're driving cross country, so I apologize for any weird we're like in the Poconos with kind of sketchy
Oh yeah,
connection,
no
but I was really
worries.
down this morning.
What's that?
So I'm really glad I was able to log on this morning, even though we have kind of a bad connection. So I want to ask you about, okay, here's where my question comes from. Big use of some digital files that are CT scans of extant bat skulls. In my teaching, I give students a long-term project where I basically give them some skulls where I tell them what species it is and I have them research the diet and figure out which characteristics are really diagnostic of that diet. And I give them skulls that are unknown to them and have them diagnose and predict what the diet might be. And I use a lot of phylostomids because the diversity is right there and we don't have to worry about some of the issues of like for connection, right? But I'm wondering, like, you had some images, must be CT scans in your talk today. Are those available for sharing, or could you talk to me more about how you get that from fossils?
Yeah, so I think we're still in the process of are you well, hopefully you can still hear me, Heather, and hopefully I caught all of your question. I think I did. I think we're still in the process of digitizing a lot of a lot of these fossil taxa. Obviously, so I just referenced the study by Camila Lopez Aguirre and a bunch of co-authors. I wasn't involved in that study. But Notonycteris, this fossil phyllostomid is out there somewhere, and I would assume that you might be able to get access to that by reaching out to Camilo and that crew. Some of the others I have started to digitize as it relates to some of my research. And actually, we just recently submitted a big grant proposal that's going to involve a lot of that if we get it. So it's really sort of to this point in terms of digitizing CT images or fossil taxa. It's been very piecemeal. I'm happy to share whatever I have if it's useful to you, but it's sort of all over the place. I think, Charlene Santana and her crew has probably done some fossil bats. Nancy may have a few more that I don't have, but it's hopefully we'll sort of be able to make a concerted effort to really CT scan a lot of these specimens very soon. And again, like I'm happy to share for your purposes in your class anything that I do have and anything that I'll be able to scan in the near future. So yeah, we're working on it. We're slowly getting there, and I think hopefully it'll start to pick up digitizing a lot of this very soon.
Yeah, awesome. Thank you for that. And well, yeah, I'll just leave it at that. We can always connect.
Yeah. No, just shoot me an email and
Yeah.
I'll let you know whatever I have that might be interesting to you.
Yeah, and then just from an educator standpoint, thank you for doing this today.
Oh yeah, no worries. I'm happy to do it. I love talking to people about fossils and bats and stuff like that. And just for anybody that doesn't know, I wouldn't be giving this talk without Heather. She was my undergrad mentor, so got me into bats. So yeah. Thank you, Heather.
Oh my gosh. Wow. I didn't realize it does like hearts now. Oh, that's cute.
Oh, wow.
I had no idea.
Yeah, anybody else with questions, I guess?
I'll throw one at you there, Matt.
Yeah, sure.
That was really great. I know very little about this stuff and love hearing and learning a little bit more about it. My question is more just about kind of the nuts and bolts of how it all happens. I mean, this all sounds to me like it's a heck of a lot of work, right? You got to get out there and get on your hands and knees, crawling around in the heat, looking for something that looks like it might be worth digging up giant piles of dirt and then hauling them all the way over and then sifting through them all. And then once you've got all that, you take it back to the lab and you got to go under the microscope to actually start seeing what you might find, right?
No
I
worries.
just wonder, what is that? Because you kind of skipped over that whole middle bit between all the
Yeah.
field work, and then we've got all this great data we can, right? So when you're going under the microscope, presumably you find lots of other stuff.
Oh yeah.
What proportion of things are bats, or what's that process look like in terms of recognizing things that you're finding when you're going under the microscope?
Yeah, yeah. No, that's a really good question, and that is sort of the most time consuming part, I think. So what we do is basically let's see. So we collect all of this sediment. We sift through it in the field in sort of panning for gold, bring it back to the lab, and then often like outsource it to undergrad workers. It's actually kind of relaxing. I enjoy doing it, but it's not something that I have a lot of time for all the time. But put in headphones and you just pick through. Most of what you're finding aren't fossils, right? You still sort of have to develop that search image. Even then, you find a tooth, and the vast majority of the time, it's not a bat, right? Like rodents are much more abundant, partly because they're on the ground, or like they're in living in ways that are much more likely to be preserved. You have things like primatomorphs, which are also just more abundant and usually bigger. So they're more likely, their teeth are more robust, they're more likely to be preserved. You can go to sites that have yielded bats before, and with enough persistence, usually find we in our upcoming grant proposal, or our grant proposal we submitted, we have proposed returning to a few sites that have yielded a few taxa of bats, and basically doing large scale screen washing and picking and sorting. We've included funding for a lot of undergrad student workers to pick through this material, because it is a very labor intensive part of the process. But once we find the specimens, the teeth fragments usually, then we can sort of compare to known fossils that are more complete, or to extant taxa. It again takes a while to develop that search image and look at something and be like, oh, this is a bat. So the undergrad or the lab technician that is picking through that material may not know that it's a bat, but at some point it gets identified by somebody who has a little bit more familiarity, or just through process of comparison with other taxa. You sort of say, okay, well, this looks like a bat, but it's kind of weird, and so maybe it's a little bit like, maybe this is a new taxon, maybe this is a new species or genus, or something like that. But yeah, so I don't know that I answered your question particularly well, but it's a very labor intensive part of the process.
Yeah, yeah, it sounds like it. No, that does answer the question. So yeah, just kind of get a sense of what it all looks like from beginning to end. And
Yeah, no, absolutely. No, picking and sorting through screen wash residue is very relaxing, but it's very time consuming, and yeah, very rarely do you find bats, but it's still cool when you find a rodent tooth or a primatomorph tooth or something like that, and like
I
there's
suspect you get along well with the folks that dissect fecal pellets underneath the microscope,
yeah.
looking at it in that way, right? Same
Yeah, it's the exact same type of thing. Yeah.
Very good, thanks.
Yeah, no worries.
All right, do we have any more questions? And actually, I think we are out of time. So we're actually going
All
to
right.
wrap it up for today in case people have other work they need to get to today. Thank you everyone so much for coming, and especially thank you to Matt for giving our talk today. And if you want to go back and like revisit anything, it will be up on the YouTube channel. Hopefully the recording didn't get too messed up with my internet failing me for a second. But yeah, and stay tuned for next month's webinar, which will be like usual, the first Wednesday of the month. For those in North America, that means bright and early, and everywhere else, afternoon, evening, and the night. So, yeah, thank you again, Matt. Thank you everyone for tuning in, and check out the YouTube if you want to afterwards. I will